This afternoon is Liam McDonnell from Leiden University, and he'll be talking about multi-imaging MS of metabolites.
Well, hello everyone. First, let me begin by thanking Tim for the invitation. So I will be talking a little bit about some multi-imaging MS of metabolites, but of course I'll give some background about how we apply multi-imaging in a clinical setting. So I'm actually a chemist by training. Everyone has given a sort of a background to themselves. Then I did a postdoc in actually a physics lab where I was doing instrument development in around late 2007. Basically I got a little bit, let's say not tired, but I wanted to apply it more. And I had an opportunity to move to this medical center and work with some really quite great people. So why do we want to do multi-imaging of metabolites? Well, one of the defining characteristics of our research is to actually always try to develop methods which can complement established, say, clinical methods. And in this case, it's imaging of metabolites. So if you think of, say, histology, the study of the microscopic anatomy of the tissue, just generally using stains, or immunohistochemistry, in situ hybridization, there are methods to image these already. Metabolites are much more difficult. There are a couple of chemical systems you can use to image a small number of metabolites. By and large, you don't have these sort of broadly applicable techniques to image a broad range of metabolites. And this is what imaging mass spectrometry can provide. So I don't know how many people are actually familiar with imaging mass spectrometry. So I thought I'd give a little bit of a background. So it's a direct tissue analysis. So imagine we have a piece of tissue here, and a pathologist colleague has sort of indicated 3 specific regions of interest. But what we can do is we can take a matrix solution, if we're looking at proteins, we'd use synapinic acid as a matrix. If we deposit a little droplet at each of these positions, this droplet will seep into the tissue, dissolving some of the proteins, or some of the biomolecules. And as the solvent evaporates, it will extract some of these biomolecules, and they'll get incorporated into the matrix crystals. And so we have these sort of matrix crystals here. This is the MALDI matrix. So when we basically shine a laser on this analyte-doped MALDI matrix, we basically generate these biomolecular ions. And of course, we've basically deposited these solutions at a very small defined region. So the molecules present in this sort of analyte-doped matrix crystal is a representation of the biomolecular content of these positions. And so when we shine the laser at this sort of small collection of matrix crystals, we generate these ions, and we can analyze these ions in a mass spectrometer. And here we see a very simplified representation where each peak corresponds to basically a different, an ion of different mass over charge. So in this case, it's idealized protein ions signals. And so what we can see here is, for example, position 2. We're missing the numbers, I do apologize, has a unique signal attached to it, but it can also just be relative intensity changes between different positions. And what's beautiful about imaging mass spectrometry is actually that the mass gives us a pretty high level of specificity. It's not quite unambiguous, that's just a little bit of exaggeration for the publication, but it gives us quite a high level of specificity. And the higher the mass resolution, or if we use SRM type techniques, or MS3 type techniques, a little bit of ion mobility, we can increase that further. We're also sensitive to different types of, say, processing, enzymatic processing. So post-translational modifications or proteolytic processing that result in a change in mass will result in 2 peaks in the mass spectrum. And so we can actually image these simultaneously. And we can apply this to multiple molecular classes using essentially the same technology, just by using different sort of matrix solutions here. And here we see basically this is some protein work at the beginning. Most of the clinical application of multi-imaging has concerned proteins. And so what we have here is a lung tumor biopsy where the pathologist has gone in and just indicated different regions according to cell type. So here we see 6 color-coded types of cells, and then these coordinates are basically transferred to a matrix spotter, which deposits a very small amount of these sort of micro droplets onto the tissue. And here we see these little sort of matrix spots on the tissue. We then analyze each of these spots, and we get a mass spectrum for each of these different cell types. And what you can see is that these different spectra from these different cell types are actually kind of different. And so we can actually look for, well, let's use the word biomarkers here, which are specific to these cell types within the tissue. This is all the direct tissue analysis. So what's a beautiful thing, one of the real strengths of imaging mass spectrometry, is that you can integrate it quite seamlessly with histology. This is actually histology defined, say, protein profiling. It's not imaging. Imaging is actually, instead of analyzing selected parts, it's actually just analyzing the entire tissue with a sort of a spatial array of spots. So again, we have the the same tissue, and instead of analyzing localized regions, we just put on a complete array of, say, these matrix droplets, or we can spray a matrix coating across the tissue, and then we analyze in this sort of spatial grid. We analyze at each position, get a mass spectrum at each position, and in the end we get this data cube of position correlated spectra, where each position, each pixel, has its own sort of mass spectrum, which is a representation of the sort of chemical makeup at that region of tissue. And we can, excuse me, analyze this in a kind of a sort of different types of methods. We can use a type of virtual microdissection where we go in again with pathology, or with histology, I mean, and we say, okay, well, we're interested in this cell type or these regions of interest, and so we can basically just take a virtual microdissection and extract these mass spectra. Or we can use different multivariate techniques, clustering techniques, or classification techniques. So here we see basically which has been probably the most common type of imaging mass spectrometry experiment. And so what we have here, so this is a prostate cancer tissue, and here we see the histological section. Here we see some imaging mass spectrometry results. And actually, after we perform the imaging mass spectrometry experiment, we can wash off the matrix and apply classic H&E stain, and then align them so we can really integrate histology and imaging mass spectrometry. And so what we can do is we can select regions on the basis of histology. Here we have the prostate cancer, here we have some benign tissue, and then we can just extract these mass spectra, and we get the, sort of what you might call the tumor mass spectrum and the benign mass spectrum, and you can just compare these sort of relative intensities of each of these peaks and look for peaks which are at much higher intensities in the tumor. And here we see one here, and then we can visualize this, and we can see that this is indeed localized in the tumor. We do this for very many tissue samples and perform different statistical tests, and we can basically identify biomarkers. And this has been performed for a very large number of tumors now. Here you see just a small number. And actually, this is actually a nice point to actually have a bit more of a discussion. Because this is the easiest type of experiment to do. You have a single tissue and you can select different regions on it. But the majority of the the results have not really gone beyond these sort of candidate biomarkers. And a pathologist might ask, well, what is this actually offering more than a regular sort of histopathological analysis if everything is already based on histopathological analysis? You have to add more. And so this is what's essential in order to get the pathologists interested. You have to have a close enough collaboration with them so that they will so you can actually decide what is a added value application. You have to get them interested effectively. And one of the areas where you can do this is to look for, say, prognostic markers that improve the differentiation or the diagnostic differentiation between various, say, diagnostically challenging tumors, response to therapy, subtyping. So in this case, this is Barrett's adenocarcinoma. It's an esophageal adenocarcinoma. And basically, imaging mass spectrometry led to the identification of multiple proteins which were associated with survival. And here we just see them here. Low expression of this S100A10 basically was associated with better survival, and the COX7A2 high expression was also was associated with better survival. Essential component, you always have to basically validate these identities. Imaging mass spectrometry is a direct tissue analysis. It's mainly of, in this case, big proteins. moldy of big proteins, it's difficult to do MSMS. They're big, they're singly charged. MSMS doesn't, or CID doesn't work too well. ETD, you don't have enough charges. And so what you can do is you can take an extract, analyze it by LC-MS, get a big table of 5,000 proteins, for example, and then what you can do is you can try a little mass matching, but moldy and electrospray are not the same. For example, we recently, we just submitted a paper where we have this one peak, we still have this one peak, 4,156 M/Z from it's actually a intestinal mucosa. And we took an extract and we performed top-down LC-MS using a top-of-the-line Orbitrap. And we identified basically more than 6,500 sort of protein fragments, non-tryptic protein fragments by top-down MSMS. Didn't have that one peak of interest. And so whenever I hear people basically saying, I've mass-matched this, if there's no verification, I'm a little bit dubious. You always have to, if we want to really apply this in a clinical setting, we have to be very, very specific. We have to be very, say, rigorous about our assignments because we want to apply this in a clinical setting. And so these are areas where imaging mass spectrometry, so all of these examples, there are solid examples where biomarkers for improved diagnostics, presence of metastasis, response to therapy, prognosis, and subtyping are already published. So imaging mass spectrometry can have an impact. Another area where imaging mass spectrometry can improve is simple multi-center studies. Most imaging mass spectrometry studies have been sort of individual enterprises and individual laboratories. So what we're doing as part of a large European network now is actually trying to compare and contrast methods and actually just internationally validate our sort of assays in different centers. But most of this is on proteins. I want to talk about metabolites because I see this as kind of a growth area of imaging mass spectrometry, especially when you consider yesterday's release of a very nice Nature Methods paper on imaging mass cytometry. If you haven't read it, I strongly advise you to. So metabolite imaging MS, not looking at proteins now, looking at metabolites. First published by the lab of Olivier Leprevotte. Like many people, they look in the literature and they see people have analyzed basically metabolites using a certain matrix. So there was a PhD student, why don't you try this? And it worked. They basically, here's a mouse brain here, they coated it with this 9-aminoacridine, and they could see a very large number of these sort of energy-rich metabolites. And then they could record the distributions here. And this has been taken up by a number of different groups, particularly some of the groups in Japan, and I'll explain why in a minute. So they started to apply it to different rodent models of neurological disorders. So this is actually not a disorder, this is actually a stroke model. And what they could actually demonstrate here for a number of different metabolites is that they would actually see changes in the metabolic distributions far before, even many hours before, any sort of histological changes were visible. And they actually started looking basically really into what's changing in these sort of metabolic pathways as a function of time following this sort of stroke model. And they pushed this further, looking with kinase-induced seizures. This is a rodent model of epilepsy. And what they could show is actually within the CA3 layer of the hippocampus, there were a very specific layer of cells which had a very, very distinct sort of metabolic signature. And again, these were basically validated using CEMS. So most of these very fine, let's say fine or high quality clinical and preclinical examples, they always include a validation step. So before we go on to this next slide, one of the reasons this is or this application of imaging mass spectrometry of metabolites is really being driven by a couple of Japanese groups is to do with post-mortem degradation. Metabolites change quickly. In Japan, they use something called focused microwave irradiation, basically to sacrifice the animal at time zero, deactivate all enzymes. And so basically they are, not in Japan, this group in Japan. And this keeps the metabolic status pristine. We're not allowed to use this technology in Europe, but there are alternatives. If you cool the animal and do basically reperfusion, a cold reperfusion, you can get the same results, but you have a much longer time window because the sort of degradation is slowed down a lot. So we first started applying this to a mouse model of migraine. And so I'll explain why we actually started to apply this. Within LUMC, we're kind of fortunate that we have one of the sort of Europe's leading migraine research groups. So migraine, most common episodic neurological disorder, affecting up to 18% of the population. It's more common in women than men. It's a very bad headache. I normally make a very bad joke here about being hungover. I'm trying to stop it. I didn't succeed, clearly. Studies of families and twins show there's approximately 50% contribution of genetics. One of the crucial parts here is it lacks clear histopathological features. And so we cannot use a histology-defined sort of virtual microdissection to look for molecular changes, metabolic changes associated with migraine. 1/3 of migraine patients experience an hour prior to the headache phase, often visual in nature. So this hour, this sort of visual disturbance in the visual field, it's actually caused by something called cortical spreading depression, which is a wave of neuronal and glial cell depolarization. It basically propagates through the cortex. And you can actually initiate or invoke CSD in a number of different ways. Mechanical stimulation, electrical stimulation, or the application of potassium chloride directly onto the cortex. And what you see then, oopsie-daisies, is this sort of slowly self-propagating wave. If you basically initiate CSD in one side of the brain, it stays in that hemisphere. And what we see, well, here we see the sort of electrophysiological sort of readings. Each of these peaks, actually I'm missing a bar here, is about 30 seconds in width. And so we applied this, we studied this by imaging mass spectrometry. This is a multi-imaging mass spectrometry with a Bruker ultraflex stream, all with the Bruker image prep. And we looked at different molecular classes just by applying different matrices. So we looked at metabolites, peptides, and proteins. And the CSD was invoked on the right-hand side. So we could see multiple peptides and several metabolites. We didn't really see anything on the protein level. There was actually a very good reason for this. Here we see the experimental design. So basically we invoke CSD, and we wait, I think it's about 5 minutes. Yes, we invoke 7 of them. So you have approximately 30 minutes between the first and the last CSD, and at this point, the animal is sacrificed. And so the time from the first CSD is approximately 30 minutes. And we know from different experiments that the peak in mRNA production following CSD is about 3 hours. So we just simply haven't had enough time to actually produce new protein, which is why we don't see any changes on the protein level here. So insufficient time for protein production. There's a traffic light here, red light. Well, hold up a minute. What about that? Means there's also insufficient time for the production of the neuropeptide precursor proteins. And actually what we think is happening is actually just that the depolarization of the cells leads to massive sort of release of the subcellular vesicles, and so the neuropeptides and metabolites are being released in that manner. So we've also started applying metabolite imaging and medicine in cancer research. This is the subject matter, also the main topic of this talk. So recall there is no immunohistochemical or in situ hybridization analog for metabolites. So this is where we can add real added value. So why is imaging metabolites important for cancer research? Well, deregulation of metabolism is a new hallmark of cancer in this 2011 paper of Hanhan and Wahlberg. And basically, in the Warburg effect, you get dysfunction in metabolism, leading to production of lactate and pyruvate after glycolysis. So basically, you have this very, very disturbed metabolism in highly proliferating cells. Also, multiple sort of metabolic enzymes or mutations in non-metabolic enzymes lead to, say, build-up of metabolites and different types of tumors. And well, the genetics of this is known because of different types of syndromes. So, for example, the IDH mutations lead to the buildup of the 2-hydroxyglutarate metabolite. This is present in glioma, central chondrosarcoma. basically in the succinate dehydrogenase, a deactivating mutation here leads to the buildup of succinate, and this is basically present in hereditary parangliomas, and similar for the fumarate hydratase. And so actually we started testing this on the FTICR, a very high mass resolution system, and we actually found that we could actually detect these metabolites, pyruvate, lactate, fumarate, and succinate. And we used internal standards, of course, to really test that we were detecting the real metabolites. And so this got us kind of excited. Okay, now we can, it looks like we can actually detect some of these metabolites. What I should say here, this is far from straightforward to perform, because these instruments have not been designed to basically scan at such a low mass range. This was an FTICR. Other instruments are designed for such a low mass range, but they don't have the same level of mass resolution. As we'll see later, each of these peaks has multiple isobaric peaks behind it. That's why we need the ultra-high mass resolution. So we then apply this to a very small patient series of triple-negative breast cancers. We analyze these by MALDI FTICR, and here we see basically this sort of mass spectrum here. And what you see is there's a severe sensitivity drop-off here. And this is simply because the mass spectrometer was never designed to go, say, below around 200. It was not the target application at the time. So we really had to sort of tune the what's a nice polite term for this? We have to tune it excessively to get down there, let's say. And if you actually do a very conservative sort of peak picking algorithm, we basically get more than 4,000 spectral features in quite a narrow mass range. And so you have this absolute sea of peaks, and you have to do something about it. It's very easy to go into this mass spectral information, find peaks which give you beautiful images. But what do they mean? The one thing I don't want to hear very much is, I have a beautiful image, I don't know what it is. This is not what basically excites our clinical colleagues. So we actually used prior knowledge. We basically go into the human metabolome database, get these metabocards out, and we just extract all of this information. And so so here we see the numbers. There are just under 3,300 unique masses, because you have different isomeric compounds. And we extract all of this information, including compound names, their exact masses, which we then converted to the M-H mass sign. It's a kind of an important aspect I think I missed off. All of these analyses were performed in negative ion mode. But we also extract the pathway membership. Some metabolites are basically in this database didn't have any sort of pathway membership. Others are involved in multiple. So we extract all of this out so we can start interrogating the data in different ways at a later stage. All of this is in MATLAB space and fully searchable and cross-linkable. And now we have a very nice little GUI to actually do this as well. So we use this metabolome database as a way of trying to clean up the data, to focus on metabolites and try to, in English we say, to sort the wheat from the chaff. I don't know if that's also a common phrase here as well. Okay. So here we see the raw data with multiple isobaric peaks, and then we just go in and say, okay, everything which is in 1 ppm, we only keep this. And so we see the database extracted. So then we can go from this to here. But this mass spectrum, every peak has also got its pathway, masses assigned to it and pathway membership assigned to it. And so then it's very easy to plot these different types of metabolic pathways and try to put images there. And I'm sure we could do this in a much prettier manner if we spend more time to it, but you get the idea. One of the things I kind of like in imaging mass spectrometry, I am a mass spectrometrist, is to annotate the tissues on the basis of the mass spectral signatures. And so we use different multivariate methods. In this case, it's a non-negative matrix factorization. I almost never use PCA. I don't like it. I could speak at length about it. I shall not. And what we see is basically, in this case, we chose 5 factors. Why did we choose 5 factors? We performed something called a corrected IKE information criterion, which gives you an estimate of which is the best sort of, say, number of factors that is the best match for your data. There's basically, as you increased the number of factors, you could match your original data better, but well, that's also always going to be the case, of course. But if you want to basically try to reduce it to a number of real factors, there's actually a penalty by increasing the numbers of factors. So this is a common method by which you can try to estimate the number of factors. There are new tools out there, such as TSNE, which is actually probably going to be better than the NMF. But I don't have time to go into that at this stage. So what you see here is 5 factors, and here we get the mass spectra in this case, and also the images, which just show basically the distribution, the intensity distribution of each of these factors. If we use factor 5 as an example, we compare that with histology. This is actually physiologically stromer. Here we see the sort of loading plot here, 152 unique masses above 5% base peak intensity. We compare this with with the database, and this can basically be mapped to 260 compounds and 64 pathways. It's because there's lots of isomers. And so if we actually had a database of real metabolites that have been detected in these tissues, we'd be able to basically improve this a lot further. So if we look at these pathways and just plot some of these, which have, say, some pathways which have, say, more, in which more than 2 metabolites were actually detected. What we do is just plot here the pathways, and this is just the number of metabolites that were detected. What you can see is basically we can start looking at this sort of pathway membership here. And actually this was also showing the citric acid cycle and androgen estrogen metabolism, and was fully consistent with basically the Warburg effect. Now, this is not quantitative, it's only the number of metabolites, but it's mass spectrometry with suitable internal reference standards. We could get sort of improvements in quantitation. Also, basically, if we have a database of absolute, of say, really detected, excuse me, or really present metabolites, we'll be able to reduce this number of sort of mapped compounds. And there was a paper that came out quite recently on nanodesi, by which actually you can do in situ MSMS on many different metabolites. And so nanodesi is basically just a liquid surface analysis of a localized liquid surface analysis of the tissue. Here you have a solvent coming in and basically interacts with the tissue in a very small region. They claim they can go down to about 10 micron diameter, and basically then you suck up, you make a liquid junction here, and there's a sampling capillary, and then this goes to the mass spectrometer. By having a very small spot size and moving the tissue very slowly, within a spot of 150 microns, they could get their sort of MS signals so you could get the the imaging mass spectrometry data, but they could also cycle through 92 different M/Z sort of isolation windows and perform MSMS. So then what you can do, you can have sort of real in situ confirmation of all of your identities, and that way you can basically reduce the number of sort of pathways that you map your data to, because you're much more confident of what the identities of your metabolites are. So actually we're currently setting this up in Italy at the moment. Improved quantitation, well, it seems a little bit mundane to say it now. It's we've heard a lot of people saying you need isotopically labeled reference standards for proper quantitation. This is also the same in imaging mass spectrometry. So in this case, it was a drug deposited onto some lung tissue. Here we see the raw data. So this is just a dilution series. And if you see this, you'd be, okay, there is a vague pattern there, but it's not overly great. The most common technique people use is something called total line count normalization. It does improve matters, but only when you add the real reference standard do you really start getting good quantitative performance. And we've actually performed this also for some of these metabolites as well. So there's C13 labeled lactate, C13 labeled pyruvate. Here we see the dilution series here. The original data was really kind of awful, but without the normalization, I mean. Or once we normalize this to the reference standard, we could get really very good or much better, say, quantitative performance. So the imaging mass spectrometry in cancer research of metabolites is really a very young area. But if you think of sort of clinical assays where you can actually complement established, say, histopathological methods, it's one area that's basically green fields. I kind of like green fields. It's easier to publish. There are challenges. If I must, talk to me if you want to know about them. I don't think we have the time thankfully. Multi-imaging MS can simultaneously trace the distributions of many metabolite ions. We can improve quantitation can be obtained using C13 labeled analogs. So we're actually working with a group in Delft now to get some of these miracle mycite subratio analysis of C13 labeled extracts. It's basically the metabolite equivalent of Silac. That's where you basically get a C13 labeled analog of all of these metabolites. And then we'll use these as reference standards to basically get improved quantitation for all metabolites. We can project the ions onto metabolic pathways, and basically this may provide new metabolite histology techniques. This is an area which has really been missing in basically pathology or histopathology research, cancer research. There is no way for them to visualize most of these metabolites. Challenges: tissue stability. This is less of a problem for ourselves, because in the hospital we use something which is colloquially termed the Jägermeister method. The Jägermeister method means everything is kept on ice. Preclinical work, you can use stabilized tissues. And one of the things I've also been thinking about is basically setting up sort of tissue-specific databases, including metabolite stability. So naturally, this is not only work from myself. In fact, I'm one of these people now, like I guess several people in here, who hardly gets into the lab. So it's basically primarily the work of Emrys Jones and Renee Van Zyl and Alexandra. Andre was my old boss. He retired last year. All of the migraine work was the people in human genetics, particularly Reinhold Scheite and Anne van der Maagdenburg. This is a beautiful survey. Anyone who speaks Dutch will know why. Anyone know what that means? Okay, this part. It means of the Virgin's Mountain. Okay. It always makes me smile when I read it. My colleagues in pathology and surgery, and naturally the people who fund the research. Thank you for your attention.
Questions? So I have one
Yeah.
for you. So in terms of time, collecting mass spectral image versus doing traditional histopathology, what's the sort of time that it takes to do one versus the other in a clinical lab? And how do you talk to pathologists about doing both or bringing that input?
It's actually a very good question. Depends on the mass analyzer you're using, the spatial resolution you want to do. So if you think of a modern MALDI-TOF instrument, maybe you can do 3 pixels per second when you're running it pretty quickly. I'm sure in the future that will be faster. I know in the future that will be faster. And so if you do 1 centimeter by 1 centimeter at 100 microns, that's 10,000 pixels, 3 pixels a second. You're talking, so what's that? Yeah, it's an hour. Not too bad. No, it's not. It's just under 3 hours. It's not too bad. If you want to go to 10 micron spatial resolution, it's going to take a lot longer. So you need much higher speed. FTICR, that data was with it's about 2 seconds a pixel, so that was longer. So that was about 150 micron spatial resolution. That's not the slow part of the data, because you can run basically 10 tissues automatically over a period of time. We've set up robots basically to feed all of these tissue samples to the MALDI-TOF simultaneously. We're not using the robot at the moment because it's not the slow point. The slow point is the histopathological annotation. This is something Rick pointed to before. Most of the imaging mass spectrometry is primarily developed in mass spectrometry labs. And so it's actually the histology or the histological annotation which is the key slow point of it. That also depends on the type of tumor you're looking at. If you look at something like gastric cancer, you get nice big regions of tumor which you can isolate. If you look at breast cancer, they're far more complicated. And then it depends on the type of breast cancer tissue you might want to look at. Because an independent prognostic factor in breast cancer is actually the ratio of stroma to tumor. And so basically when you start getting this sort of fine sort of distribution of stroma throughout the tumor, they have a much worse prognosis. but people don't like to really analyze them because the annotation gets very detailed. And so people a lot of the earlier studies were focused on these sort of nice tumors which have nice regions that you can easily isolate. One of our projects, this one, differentiation and characterization of clinically challenging soft tissue sarcomas, they're diagnostically challenging. I didn't realize how diagnostically or how histologically challenging they were. When I wrote the proposal, I was reliant on my pathology people. The PhD people, or PhD students, were looking at these, and it was like, well, how do I know what I'm looking at? Our pathologists were saying, well, you need 10 years to understand it. These are enormously complicated. There's more than 50 subtypes, all with overlapping histology. So how long is a piece of string? It depends what you're looking at. You have to basically, you need good interactions with pathologists because you also need the clinical information.
Yeah.
Yeah.